Bom, pessoal, então sejam todos bem-vindos novamente a mais um encontro nosso aqui, né? Da nossa disciplina, só um minutinho, da nossa disciplina de controle da plasticidade e função muscular esquelética, que é oferecida pelos programas de pós-graduação em ciências do esporte da UFMG e de ciências fisiológicas da UFTM. Meu nome é David Gonçalves, sou professor da UFMG, responsável pela disciplina, e aqui comigo o professor William e Assis Silveira, da UFTM.
Olá, pessoal.
Está conosco. Isso aí, William. O pessoal vê a gravação, está aqui acontecendo, tudo certo. E a gente, então, hoje, dando sequência nas palestras que fazem parte dessa disciplina que a gente tem oferecido, lembrando que nós temos, já vou lembrar aqui no início, mais 2 encontros, né, nas próximas 2 semanas. Então já fica o convite para todos. Quem tiver interesse, só fique atento, porque os horários eles vão alterando aqui nas semanas, conforme a disponibilidade, o fuso horário dos palestrantes. Então a última do Dr. Bert Pelão, por exemplo, vai ser mais cedo. Então acompanhe, por favor, entre no site do Lafise, do Laboratório de Fisiologia do Exercício aqui da FMG, né, ou acompanhe no Instagram também, do Lafise, onde essas informações estão sendo postadas semanalmente. Bom, hoje a gente tem o prazer, o imenso prazer de receber aqui conosco o professor Dr. Brian Neal, da Universidade de Iowa, nos Estados Unidos, e ele vai vir apresentar o seu trabalho, então, e discutir um pouco conosco. Então, assim, é um prazer realmente enorme tê-lo aqui conosco. Feito essa pequena introdução aqui, rapidamente, em português com vocês, eu vou agora pedir licença para que a gente possa, então, falar inglês, para que o professor Brian possa, então, entender, né, bem o que a gente está falando, embora eu já vi aqui na aula anterior que ele já estava pescando algumas coisas que a gente estava falando, então, acho que está acostumado com o espanhol, mas já pegou alguma coisa aqui. Mas então vou mudar para o inglês aqui. Sejam todos bem-vindos. E só vou lembrar a configuração, ele vai fazer a apresentação agora, 30 a 40 minutos, e no final nós teremos aproximadamente 20 minutos para discussão. Tá? Então agora eu vou mudar para o inglês só para... So, Brian, I introduced our class and your lecture for everybody in Portuguese. So now I have to say that I'm very pleased to introduce you for everybody. We first met each other in 2015 during our postdocs. Dr. Brian was in the Dr. Ronald Kahn's lab in Joslin Diabetes Center from Harvard Medical School, and in that period. And I went there to perform some experiments in muscle-specific insulin receptor knockout mice. And Brian helped me a lot, and he was really, really great. So thank you very much again for helping me that moment. And now to be here. So Dr. Brian got his MD and PhD degrees in biochemistry at the University of Utah in the United States, and did his postdoc at Harvard Medical School, as I told before. And now he's the assistant professor of internal medicine in endocrinology and metabolism in Fraternal Order of Eagles Diabetes Research Center from the University of Iowa. And so, Brian, thank you very much for being with us today. Thank you again.
Yeah, of course. I'm very proud to be here, and thanks for the very kind introduction. I'll start sharing my screen just to...
Okay, yeah, you can share your screen now. And after the presentation, we hope to have 15 or 20 minutes to discuss about some questions. So please, guys, you can write here in the chat your questions, or just ask them. So, Brian, thank you again, and you can start your talk when you want.
Well, again, thanks very much for the introduction. I am very happy to be visiting Brazil for at least an hour this afternoon. Hopefully in the future we will be able to do these in person. And I'm very proud to be able to talk to you about the work that my lab has been able to do over the past 6 years since I moved from Boston. I'm going to touch briefly on some of my postdoctoral work, but hopefully spend most of the time on our recent work. And that work really describes the multiple roles of FOXO signaling in diabetic muscle disease. I wish I could tell you that you should come to Iowa to see such beautiful rivers and mountains, but this is not Iowa. This is actually New Mexico, a different state in the United States in the Southwest, and a very beautiful place to visit. But I certainly invite you to the University of Iowa. I do love it here, and anybody who would like to come and see our state would be wonderful. I'm going to have 3 objectives for this talk. And please, if anybody has questions in between time, just unmute yourselves and let me know. I think in the current format, I can't actually see the chat or anything, or even raised hands. So please feel free to unmute yourself and ask a question. But my objectives are going to be to briefly describe the clinical impacts of diabetes on skeletal muscle function. I'm going to describe to you the dual roles of FOXOs that we have identified in diabetes-related muscle atrophy and in mitochondrial dysfunction. And then I'm going to spend a little time at the end to present the transcriptomic data that we have on what insulin action and IGF action and the FOXO isoforms and how they may have a role in calcium homeostasis. So I am a clinician. I see patients with diabetes. And if you go to Google and look for diabetic complications, as I did when preparing this talk, you will find a picture similar to what's written here. Multiple different parts of the body are affected by diabetes. And this hits all the major ones that I warn patients about, what are called microvascular complications, including eye disease, kidney disease, or even foot ulcers and nerve disease. The thing that actually imparts the most mortality risk is the heart disease of diabetes that can lead to heart attacks or strokes. But if you look beyond the sort of large ones that I've highlighted here, you see that diabetes also has impacts on mental health, on the mouth in periodontal disease, so how the teeth work, sexual dysfunction, lower limb problems. What you will notice is absent in this picture is the skeletal muscle. Now, most of us in physiology classes learn that muscle is very important for insulin signaling and or glucose homeostasis, but this says that diabetes doesn't actually have an impact on the muscle. But I'm going to show you some data that it does have a critical impact on skeletal muscle function and strength. This is a study, some data from a study that I'm showing you that will eventually conclude older patients with diabetes lose muscle strength faster. So this was a study done in what I like to call free-range humans, people living out in the community, and they were elderly, 70 to 80 years old. What the study did was it took these patients, measured body composition and muscle strength, and then measured it every year for a total of 6 years. Not surprisingly, if you didn't have diabetes and you were 70 to 80 years old, you did lose some muscle strength over the 6 years, about 1.2 Newton meters per kilogram. I tried to estimate this out. I think it was about 5 pounds on a leg press that would have been lost over the 6 years in these 70 to 80-year-old patients. If you had diabetes, you lost it about 30% faster, even without any big change in the diabetes control or things like that. If your diabetes control was poorer, you lost muscle strength even faster than that. So this right here tells us that diabetes somehow affects muscle strength in older individuals. A second piece of data that's probably much more important to my patients with diabetes is what happens after a major surgery. That is a time when muscle strength declines rather rapidly due to being bedridden or more inactive. This was a study where they did hand grip strength, so nothing related to the actual heart, after a coronary artery bypass graft, basically an open heart surgery. If you did hand grip strength 1 month after the surgery, patients with diabetes had a certain amount of hand grip, or patients without diabetes, patients with diabetes had about 25% less strength. And these two observations are very important even for mortality. Within this study of nearly 1,800 people, they also looked at death rates among patients based on their strength. And the overall conclusion of the actual study was that skeletal muscle weakness is strongly associated with mortality in humans. What we see is that if you measure everybody's strength and line them up from the strongest on the top to the weakest on the bottom, and then divide them into 4 groups, the top group had a certain mortality of about 92% over the 6 years of time. That may sound like a lot, but remember, these are patients who are 70 to 80 years old. If your muscle strength was in the next third or the next fourth, you had an increased rate of dying, and then the people who were in the bottom level of strength, so the bottom fourth of strength, had almost a 35% chance of dying over the course of this 6-year study. And so what I hope this data shows you is that diabetes is somehow related to decreased muscle strength, and that's important for mortality. I put a double-headed arrow here because we don't know if diabetes causes decreased muscle strength, or some of the things that go along with diabetes decrease muscle strength, or perhaps even the decreased muscle strength contributes to diabetes. There are a number of factors that could influence this relationship. I've divided them into metabolic factors. Those would be impaired insulin signaling, hyper or hypoglycemia, meaning high or low glucose values, blood sugar, or dyslipidemia, changes in the lipids, or there could be other disease processes. I just told you that patients with diabetes have a high risk of heart disease and heart failure. There's peripheral vascular disease that would make it tougher for the muscles to actually contract, or the neuropathy that goes along with diabetes. If the nerve can't get the signal down to the muscle, the strength is going to be affected. With all these various potential contributors, and again, I've only listed 6 of probably hundreds that are associated with diabetes, we wanted to ask the question, what does impaired insulin signaling alone do? And we developed mouse models to try and test if you impair insulin signaling and or its very closely related receptor, IGF-1, what does that do to muscle strength? To do this, we developed mouse models with efficient recombination of insulin receptor, which I've denoted IR, and IGF-1 receptor, which is IGF-1R, in the skeletal muscle. So to do this, we take a mouse that has Cre recombinase under the ACTA1 promoter. Here shown very restricted to skeletal muscle. It does get a few heart cells, but not enough to be significant. And we crossed it with the insulin receptor flox mice and the IGF receptor flox mice. Our groups were controls that had no Cre. They have normal insulin and IGF receptor expression throughout the body. Muscle-specific IR knockouts, these lack insulin receptor just in skeletal muscle. IGF receptor muscle knockouts, or what I'll sometimes refer to as the MIGRCO or double knockout, muscle IGF receptor insulin receptor knockout. Now we wanted to find a model that didn't have all the other complications of diabetes. So the first thing that we did was to ensure that when we knock out the insulin and IGF receptor in muscle alone, it doesn't actually cause diabetes. Here's a glucose tolerance test where we fast the mice overnight and then give them a load of glucose and see how they handle it. You can see that the McGurko mice, although they had lower blood glucoses after a fast, their glucose excursions, or their ability to clear the glucose from their blood, was the same as controls, indicating they don't have diabetes or even a form of whole body insulin resistance. We did confirm that the insulin and IGF receptor were actually knocked out, and there was no insulin action in the skeletal muscle. But what was more obvious to us was the muscle atrophy. Here on the left is a control mouse hind limb. You can see a nice bulk of skeletal muscle in the quadriceps, quadricep of here, hamstring, and gastrocnemius muscles. In the McGurko mice, they had severe muscle atrophy. We quantified this we quantified the muscle atrophy both by showing dissected muscle weights and also grip strength. And what you can see is that if you just lose insulin receptor, showed in the hatched bars, you get a small decrease in tibialis anterior muscle, extensor digitoris longus muscle, and not as much in the soleus muscle. IGF-1 is much more known for its muscle hypertrophic effects and has been described in the literature as being a critical regulator of muscle size, and yet we knocked out IGF-1 receptor and see no difference in muscle size in any of the groups that we tested, muscle groups. However, there is some overlapping role of insulin and IGF receptor, because if we knock out both in the McGurko mouse, shown in black, we get a severe muscle atrophy, almost 60%, which is obvious just to the naked eye, as shown up here. And these decreases in muscle size correlate with decreases in grip strength measured in the mice, such that insulin receptor causes a mild decrease, loss of both receptors causes a marked decrease in muscle strength. This was a majority of the work that I did during my postdoctoral fellowship, and it was published in JCI in 2016. And to summarize a large majority of that work, I had to teach myself about the molecular mechanisms for muscle atrophy and hypertrophy. And of course, these are a balance between protein synthesis and degradation. We found in the McGurko mouse, which had severe muscle atrophy, that there was a paradoxic increase in protein synthesis and the synthetic pathways that contribute to protein synthesis. But this was largely due to a large increase in protein degradation pathways, causing a high protein turnover state. When we looked in the McGurko mice at proteasome and autophagy lysosome pathways, depicted here, we found they were highly upregulated in the McGurko mouse. And we were fortunate that we developed this model in about 2013, and about a decade previously, FOXO transcription factors have been demonstrated to be the critical regulators of these two pathways in skeletal muscle. Why were we fortunate? Because if we simplify the insulin-IGF receptor signaling cascade, we find that these three isoforms of FOXO that are expressed in muscle are controlled by insulin and IGF action. That is to say that when insulin and or IGF are present, they signal through PI3 kinase and AKT, they phosphorylate FOXOs and suppress their activity. We were able to show in our mice that when we deleted insulin and IGF receptors, the FOXOs translocate to the nucleus and activate atrophy and autophagy genes. This was a very fortunate pathway for us, because once we deleted two receptors, we activated transcription factors. This allowed us to actually delete the transcription factors to see if we could rescue the phenotype. And after a very long experiment in mouse breeding, we were able to achieve deletion of FOXO 1, 3, and 4 in the McGurko background. So shown here are Western blots of all the proteins that we were talking about. In controls, they have normal expression of insulin receptor, IGF receptor, and the three FOXOs. When we knock out insulin IGF receptor in skeletal muscle, we see that insulin receptor is completely gone. IGF receptor is actually highly expressed in fibroblasts and pre-myoblasts, which seem to go up in this model, trying to likely compensate for the atrophy. But FOXO 1, 3, and 4 are normally expressed. We had our control where we knocked out all three FOXOs, 1, 3, and 4, and we have what I call the muscle quintuple knockout. That's 5 different genes. Insulin receptor, IGF receptor, and FOXO 1, 3, and 4 are knocked out. Well, what happened to the muscle? Shown here, you can see that it completely rescued the atrophy phenotype that we observed. Control shown in white, as far as quadriceps muscle weight, was normal. When we knock out insulin and IGF in the McGurko, shown in black, we get that 60% muscle atrophy. When we knock out the three FOXOs, we actually didn't see a difference in muscle size in this early age, about 8 weeks. But when we knock out all 5 genes shown in blue, we see that the quintuple knockouts have normal muscle size, and for all intents and purposes, look like a normal mouse in the cage. Interestingly, this also reversed, or I guess confirming our hypothesis, this also reversed all the changes in autophagy lysosome and proteasome degradation. Shown here is LC3, a critical regulator of autophagy pathways. It was accumulated in the McGurko mice due to dysregulation of both activation and likely impaired clearance of autophagosomes. This was completely normalized in the muscle quintuple knockout. This is a very interesting genetic trick, is what I would say. There are no humans out there that lack insulin receptors or IGF receptors just in their skeletal muscle. And so the next question we had to ask was, well, how do these mouse genetic tricks that we've done, these investigations of insulin and IGF signaling, relate to human disease? To do this, we actually decided to study the FOXO triple knockout mice and make them diabetic. So here I'm showing a separate set of experiments that we've since published in diabetes. And what you can see is that when we give the mice STZ, it's called streptozotocin, it kills off the mouse's beta cells or insulin-producing cells, and they get a form of type 1 diabetes. Here shown the random blood glucose levels in the control mice are within the normal range for a mouse. When we give streptozotocin, we get frank diabetes with glucose levels of 500 to 600 milligrams per deciliter. To convert that to millimoles per liter, this is somewhere in the 20 to 30 range, or even above that. We also did this in our muscle-specific FOXO triple knockout mice. So remember, these have normal insulin and IGF receptor expression, but they lack the FOXOs in skeletal muscle. When we treat them with STZ diabetes, they have the normal response and become just as diabetic. What the FOXO triple knockout mice don't do is develop any muscle atrophy. So again, the STZ diabetic model shown in gray has about a 20% decrease in muscle size in quadriceps, TA, and EDL. Basically in any muscle group that we looked at. But when we knock out FOXOs, the diabetic group did not show a significant decrease in nearly any muscle group that we had. To equate this to humans, we actually did some qPCR, some mRNA levels of the critical atrophy genes. And we found that the FOXO-dependent atrophy genes that are increased in mice and type 1 diabetes in humans are going to be shown here. So just to orient you again, control mice in white, the STZ diabetic mice in gray, we find that critical regulators of ubiquitin proteasome degradation, including FBXO32, which is also known as atrogen-1 or MAPBAX, this transcript goes up with diabetes. It is completely prevented when FOXOs are knocked out. TRIM63, also known as MERK1, has the same pattern, and several of the autophagy genes, such as BNF3, BNF3L, and GABARAPL, all increase with diabetes and are prevented with FOXO knockout. We were fortunate to collaborate with Dr. Sri Nair at Mayo Clinic and actually get some muscle biopsy samples from patients that had type 1 diabetes. And the unique thing about this study was that these patients came in and had a muscle biopsy when they had normal insulin treatment. So we give them insulin, control their blood sugars, and did a normal muscle biopsy, or actually had all of their insulin withdrawn for up to 8 hours in a controlled research setting, what I call insulin deprived, shown in blue. And what you can see is that the muscle biopsies from the patient when they had normal insulin, compared to when they had insulin deprivation, we get a significant increase in several of these FOXO-mediated ubiquitin proteasome genes, as well as a couple of the autophagy lysosome genes. And so we hope that this helps prove our initial hypothesis. And that is that in the context of muscle growth and keeping normal muscle mass, insulin and IGF receptor signaling is critically important. And this is mediated through the FOXO transcription factors. The suppression of their activity allows for growth and maintenance of muscle mass. By contrast, under fasting conditions shown by other groups and diabetic conditions shown by our group, we see that FOXOs become activated. This activates the protein degradation pathways, both ubiquitin proteasome and autophagy lysosome, and can lead to atrophy in a high protein turnover state. So now I want to shift gears to what my lab has done more recently, and this is to investigate energy production in the context of insulin or IGF receptor signaling. As many of us know in skeletal muscle, once the nerve activates the muscle cell, the T-tubules allow for calcium release from the sarcoplasmic reticulum, and this triggers contraction both through the actin-myosin coupling. ATP is highly needed for muscle contraction, both to induce the contractile process, but also to pump the calcium back into the sarcoplasmic reticulum through the Serco pump. The muscle, unlike the heart, uses a bit more glucose for its fuel substrates. And certainly under anaerobic exercise conditions, it relies almost entirely on glucose and glycolysis to produce ATP. But under normal resting conditions, either needed for normal posture, standing normally, or even just walking, or even aerobic exercise, the majority of the ATP is produced from the mitochondria from both glucose and fatty acid substrates, as well as amino acid substrates. We wanted to investigate the mitochondrial function in the context of diabetes. And actually, it's been well studied over many decades. Here is just one study from our collaborators in those insulin-treated and insulin-deprived type 1 diabetic patients that shows that if you just withdraw insulin for about 8 hours, you get a reduction in ATP production from the mitochondria. What they did was, with the muscle biopsies they initially obtained, they isolated mitochondria from them, and then they fed them various substrates, shown on the bottom, and I'll describe what these substrates are, and measured ATP production. What you can see is that in the insulin-treated phase, they had a normal amount of ATP production with pyruvate and malate as substrates, or glutamate and malate, or a mixture of pyruvate, palmitoyl carnitine, alpha-ketoglutarate, or N-malate. In every case, there was a significant decrease in the ATP production by the mitochondria in the insulin-deprived state. However, this still doesn't tell us what are the cellular signals that mediate this decrease in ATP production. One thing I like to teach our fellows about, and teach a lot of people about, is that mitochondrial structure is very different in muscle than a lot of other places in the body. Here I'm just showing you a essentially Google image of a skeletal muscle fiber. Here's a single skeletal muscle cell. This is polynucleated. It's much longer than what you see on the screen. But the pink elements are the contractile fibers. The purple and blue lines here are the T tubules and the sarcoplasmic reticulum. And unfortunately, mitochondria, as they are normally drawn in textbooks, look like little beans in the cytoplasmic mix soup. These cyan things are meant to be the mitochondria. Unfortunately, this is not how mitochondria look in skeletal muscle. Here was a report from Dr. Brian Glancy in Nature of 2015, essentially showing that the mitochondrial structure is very complex. What he did was he took electron micrographs that were serial sections, so multiple sections in a row, and used computers to reconstruct a 3D image from these serial electron micrographs. Here's a muscle fiber that is much larger than what you see. It would be completely filling the entire screen going in and out to the left. What you see here colored is a blood vessel shown in red and pink, a nucleus of the skeletal muscle shown in blue, the contractile elements all shown in gray, and the mitochondria shown in green. With this computer image, we can actually take away the contractile elements and see these long, intricate sort of networks of mitochondria that go through them. If you were the first one to do this, as Dr. Glancy was, you get to name all of these types of mitochondria and use all the various acronyms that you would like. So he called these paravessel mitochondria, intramyofibrillar mitochondria. He then coined these doublets as I-band mitochondria, since they seem to run right along the I-band of skeletal muscle. There's cross-fiber mitochondria, fibered parallel mitochondria, and again, just this intricate network of mitochondria within the skeletal muscle. Traditionally, the way to test mitochondrial function is to grind up the tissue using a mortar pestle homogenizer, and then through 2 different rounds of centrifugation, you get the mitochondrial pellet out. And if you do this, you actually get the beans that we talked about before, this globular shapes of mitochondria. However, there's another way in skeletal muscle to test mitochondrial function, and that is to individually separate the muscle fibers from each other, and then treat them with a saponin, which is a detergent that is rather specific for the plasma membrane. And you can see that the mitochondrial structure is intact when we do this. Either method that you use, you can then test the mitochondrial function if you have an oxygraph, a high-resolution oxygen sensor. We use the Ouroboros oxygraph in our lab, and we are able to give these fibers or mitochondria pyruvate with malate and measure oxygen consumption. We can also give palmitoyl-carnitine, a fatty acid substrate. We can give glutamate as an amino acid substrate, or we can directly test the electron transport gene, the oxidative phosphorylation, with succinate. And all the while, we can measure oxygen consumption, and using a separate machine, we're actually able to measure ATP production. So I want to show you some of the data that we've generated in our various mouse models. So using the saponin permeabilized fiber technique, where we have intact mitochondrial structure, we gave pyruvate and malate to our insulin receptor knockout mice alone. Remember, these have very mild muscle atrophy. They have normal expression of IGF receptor and FOXOs, and if we see the oxygen consumption upon maximal stimulation with ADP is mildly but significantly decreased when you just lack insulin receptor. After succinate, rotenone, and oligomycin and FCP, we see much less of a defect in the mitochondrial function. This would suggest it's more restricted to a complex one of the OXPHOS, but we'll come back to that in a few minutes. When we take our double knockout mice, remember the severe atrophy McGurko mouse, we find a severe defect in mitochondrial function. We've now got nearly a 60% loss of mitochondrial activity or respiration in these individual fibers. And remember, this is all normalized per milligram of soleus, so even with a smaller muscle, the mitochondria are even doing worse than that. We are able to recapitulate both of these in isolated mitochondria using the grinding technique, but really the role of FOXOs was illuminated when we did this in our quintuple knockout mice. So when we knock out insulin, IGF receptor, and all 3 FOXOs, we get normal muscle size again, and we get normal respiration again. Again, we wanted to know, does this actually occur in the diabetic state? And is this downstream of FOXOs? So using isolated mitochondria, we again made the mice diabetic with streptozotocin. Now I'm not showing you, but the mice again had very high glucoses in this experiment when they were given STZ. The control mice that didn't get diabetes have a normal amount of respiration. The STZ diabetic mice have a significant decrease in both their oxygen consumption as well as their ATP production. But when we lack FOXOs in the skeletal muscle, we don't see this significant decrease in respiration or in ATP production. The next question was, how do FOXOs modulate the energy production? FOXOs have been well characterized to target those genes for protein degradation, but have been less well characterized as far as their influence on mitochondrial proteins. Well, perhaps it wasn't a targeting of mitochondrial proteins at all. Our first clue came from this paper published in 2018 in Diabetologia in the lab of Dr. Perry, where they did muscle biopsies of patients with and without diabetes. And you can see that with diabetes in black has a significant decrease in their oxygen consumption at various levels of ADP. And in those muscle biopsies, they actually looked at the mitochondrial structure. In the controls, shown in red here, the mitochondria looked quite healthy with normal cristae. In the type 1 diabetic individuals, the mitochondria looked rather oblong, and these blue structures here were what they described as remnant autophagosomes. Perhaps that mitophagy was contributing to this. So what is mitophagy? Well, autophagy is the process of engulfing a component of the cell in a double membrane structure and degrading it in the lysosome. Mitophagy is merely the engulfment of mitochondria and degradation through the autophagy lysosome system. Since we already demonstrated that FOXOs control autophagy, this was the logical experiment to do. We used a fluorescent tag called mitotimer. This fluorescent probe is tagged to mitochondria. It is initially green, and then as they get oxidized, it eventually turns red. And it has been shown that this mitotimer, when the mitochondria are localized in the lysosome for degradation, it is completely red. With this mitotimer plasmid, we also tagged the lysosomes in yellow to see the co-localization of red mitochondria with yellow lysosomes. What we found was in our control mice, we could co-localize an occasional red dot with the LAMP1 positive lysosomes. So the mitochondria are co-localizing with the LAMP1 YFP, but a majority of the mitochondria were not. All the other reddish yellow hue here is not localizing with the lysosomes. This is in the inducible double knockout, which also has a strong mitochondrial phenotype. We could also locate occasional mitochondria co-localizing with lysosomes. But when we quantified all of this, we actually found no difference in the oxidation rate of this mitotimer and no difference in the number of mitochondria in the lysosomes. So if you read the title, you got it right at the beginning. Mitophagy is not activated in the insulin IGF receptor knockout. So if mitophagy is not the answer, what else could be leading to these decreases in oxygen consumption, ATP production, and reactive oxygen species? Well, we knew that FOXOs are transcription factors, and so we decided to look for the mitochondrial components and the electron transport chain subunits to see if they were regulated by FOXOs. So we did an RNA-seq experiment, and perhaps not surprisingly, we found that the insulin receptor knockout mice mildly affect the transcripts. If we do a rather stringent statistical cutoff here, we find that only about 23 genes are changed in the insulin receptor knockout, only about 11 genes are changed in the IGF receptor knockout. But even with a very strict cutoff of an false discovery rate less than 0.01, the Gerco mice had a huge number of genes that were statistically changed. When we look at the pathways in this Gerco mice, we were encouraged to find that among the top 10 was the mitochondrial dysfunction pathway. Shown here are the number of genes within the pathway that are up or down regulated. And what we found rather unique in the mitochondrial dysfunction pathway was that a majority of them were down regulated and almost none were up regulated. We next wanted to compare, is this due to FOXOs? And so we tested again in the muscle quintuple knockout, and we find that a majority of the transcriptional changes that are seen in the McGurko mouse are actually reversed in the quintuple knockout mouse. And if we ask, of all the genes that are FOXO dependent, what are the pathways that are most regulated? It turns out oxidative phosphorylation is right at the top, and it is down regulated, as shown here by the blue versus the orange. The second mitochondrial pathway in these top 10 was the Tca cycle, which was also down regulated. This was helpful because these are two of the major pathways that are required for normal mitochondrial function, as shown in the picture here. So let's start with the Tca cycle. We pulled out several of the genes from the Tca cycle and found that indeed they were all down-regulated in the McGurko mouse and normalized in the MQKO mouse. We actually did some metabolomics and identified that the metabolites just downstream of many of the genes that were downregulated have accumulated. So this blue means that this is accumulated in the McGurko mouse relative to the others, and not accumulated in the QKL mouse. We found cisaconate, malate, fumarate, and succinate. All of this means that a significant downregulation, at least the transcriptional level, of the Tca cycle metabolites led to an accumulation and a blockage of this pathway. The most regulated pathway was the electron transport chain, also known as the OXPHOS or oxidative phosphorylation cascade. Shown here are the 5 complexes that comprise the OXPHOS electron transport chain. Complex 1 oxidizes NADH, and the protons are pumped at the various sites and eventually flow back across in complex 5 to generate ATP. This is many more genes than were in the Tca cycle. Here's just a small list of all of them. You can see that complex 1 has nearly 44 genes associated with the subunits that make up this complex, whereas complex 2 is the smallest at 4. We extracted the RNA-seq data from our McGurko mice and our controls, and they're all shown here. What you can see is that complex 1, which is shown in this and this graph, had a significant downregulation in a majority of the genes. But many of the complexes had other genes that were downregulated, that were shown in red compared to the normal expression in blue. What we were encouraged to find was that in the quintuple knockout, a number of these genes, shown by the stars, were statistically normalized in the quintuple knockout mouse. What was interesting was that there was a large proportion of the complex 1 core subunits, and this is the core subunits are the ones that are conserved all the way back to archaebacteria and comprise the 2 core catalytic sites of complex 1. We find that when we measure the activity of complex 1, 2, and 3, that this mimics a lot of the gene expression changes. So in McGurko, complex 1 activity is markedly decreased. Complex 2 activity, which had one of its 4 subunits downregulated, was also decreased, but complex 3 was relatively unchanged as far as its enzymatic activity. In the QKO, again, each of these complex activities was normalized. So I'm coming towards the end here, and I hope that I have convinced you that within skeletal muscle, insulin and IGF play a critical role in maintaining mitochondrial function. And in the diabetic state, where we have shown downstream of insulin and IGF receptors, FOXOs become activated. They lead to mitochondrial dysfunction, which is characterized by decreased respiration, decreased ATP production, and what I didn't show, we also saw increased reactive oxygen species. And if we look more detailed at the regulation by FOXOs, we see that complex 1 is more highly affected than the other complexes. And even super complexes, which I didn't show you, are affected by these defects and transcriptional changes by FOXOs. And all of this was just recently published within the last couple of months by my talented postdoctoral fellow, Dr. Bardawaj, in the Journal of Clinical Investigation. And we think that this is at least a contributor to the decreased strength seen when insulin and IGF signaling are disrupted. So a question I often get is, why don't we just make selective FOXO inhibitors to improve muscle function in diabetes? I've just shown you that FOXOs seem to control mitophagy, autophagy, a lot of metabolism, protein homeostasis, but this is just one piece of the FOXO pie, as I like to say. And actually, FOXOs do a lot of regulation of oxidative stress and even growth factor repair, differentiation, DNA repair, and cell cycle arrest. And the unfortunate thing is that if you knock out FOXOs outside of skeletal muscle, or if you inhibit them outside of skeletal muscle, here's just a mouse model that knocked out FOXOs within the uterus of mice or in the bone marrow, you actually get tumors and cancer production. And so we need selective FOXO inhibitors. We need FOXOs, we need to know what FOXOs interact with to impart their metabolic and protein homeostatic effects and try to avoid the cancer-causing effects of inhibiting FOXOs. So again, just to summarize, we've shown that insulin action suppresses FOXO activity, and when this is unrestrained, we see a dual role for FOXOs, both inducing protein degradation to cause muscle atrophy and inhibiting mitochondrial function, which could also lead to decreased muscle strength. And we hope that our investigations in the future will identify the targets of FOXOs that mediate these effects and allow us to develop better therapeutics. So with that, I need to thank the members of my lab, Christy Penniman and Dr. Bardawaj, who've been with me for the past 5 years. Dr. Bardawaj just recently published his JCI article. Had a number of excellent, talented postdocs and undergraduates in my lab, some top-notch collaborators, and must thank the Veterans Affairs for our funding, as well as the support from the Fraternal Order of Eagles Diabetes Research Center, which I'm a part of. With that, I'd be happy to take some questions.